All right, welcome to our deep dive. And today we're going to be talking about viruses.
Oh,
It's
cool.
going to be like a biology class, but don't worry, no tests.
Awesome.
So get ready to learn all about these fascinating little things
I'm excited.
that play such a huge role in, the world around us.
Definitely.
So when we think about viruses,
Uh-huh.
I think, a lot of times we think about the negative stuff,
Yeah, yeah.
like, diseases they cause.
Right,
But
right.
today we're going to go beyond that
Absolutely.
and really kind of explore, like, what makes them tick.
I like that.
How they work, how they replicate,
For sure.
and even some of the ways that they're actually being used for good,
Yeah, that's a cool part.
which is something that I didn't really realize until I started doing a little bit of research for this.
Definitely, yeah.
So to kick things off, I have a question for you.
Okay.
Are viruses alive?
Oh, that's a great question. And it's one that has puzzled scientists for a long time. So, to be considered alive, an organism needs to have a cellular structure.
Okay.
All those tiny factories and compartments we see in diagrams of cells.
Right, like the cytoplasm and organelles and all that stuff.
Exactly. But viruses, they don't have any of that.
Really?
Nope. They're basically just genetic material, either DNA or RNA,
Huh, okay.
packaged inside this protein coat called a capsid.
So like a little suitcase for genes.
That's a great analogy. They're like little genetic nomads drifting around until they find a cell to hijack.
Uh-huh. So that's how they spread and cause disease, by hijacking our cells.
You got it. They can't reproduce on their own.
Oh, so they need our cells to do it for them.
Exactly. They need that host cell's machinery.
Sneaky little things. But wait, if they're not technically alive, how can they do all that? How do they know which cells to target?
Well, they've evolved this incredibly efficient strategy. Their proteins act like keys fitting only into specific locks on the host cell's surface.
Oh, so they're picky about which cells they infect.
Very picky. For example, the T4 virus, which you can see in figure 11.7 in the textbook,
Oh, yeah, I see it.
infects only certain bacteria. It can't touch plant or animal cells.
Wow, so it's all about finding the right fit. So, okay, once a virus finds its match, what happens? How does this hijacking actually work?
Well, that's where the lytic cycle comes in.
Lytic cycle.
Yeah. It's a multi-step process, but we can break it down.
Okay, step by step. I like that.
Perfect. So first, imagine the virus docking onto the host cell, almost like a spaceship.
Gotcha.
That's the attachment phase.
Okay, attach.
Then it injects its genetic material, either DNA or RNA, into the cell.
Like sneaking in the blueprints, huh?
Exactly. It's like, here's how to make more of me.
And what happens to the protein coat, the capsid?
Good question. That stays outside the cell. Only the genetic material gets in.
Interesting.
Then, using the host cell's own machinery, the viral genetic material starts to replicate and translate into new proteins.
So the cell is basically turned into a virus factory.
Exactly. It's a very clever strategy.
And then,
Well, next comes the assembly phase, where all these newly made viral components start coming together, forming new virus particles.
wow,
And finally, in a grand finale,
what happens?
the host cell bursts open, releasing all those new viruses to go infect other cells.
Oh, wow. So it's like a microscopic explosion.
You could say that?
No wonder we feel so lousy when we're sick. Our own cells are being hijacked and destroyed. So is this lytic cycle the only way that viruses reproduce?
It's one way, but not the only way.
Oh, tell me more.
Some viruses are a bit more subtle, opting for a stealthier approach called the provirus stage.
Provirus, is that like a pro at being a virus or something?
You could sing that. Instead of immediately destroying the host cell, the viral DNA integrates itself into the cell's own chromosomes.
So it's like going undercover, hiding out in the cell's DNA.
Exactly. And the host cell, none the wiser, replicates the viral DNA along with its own every time it divides. So every new cell produced also carries a copy of that provirus.
Wow, so the virus is replicating without causing immediate damage. That's a pretty long-term strategy. Are there any examples of viruses that do this that, like, we might know?
Oh, absolutely. Think about like the herpes simplex virus, the one that causes cold sores.
Oh, yeah.
Those sores pop up when the virus is like actively replicating,
Uh-huh.
but then they disappear, right?
Right.
That's the provirus like hiding out, lying dormant in the cell's DNA.
Ah, so it's like a viral sleeper cell.
Exactly.
It makes sense now why those cold sores seem to just, like, come and go.
Yeah.
But if the virus is just kind of hanging out there in the DNA, what triggers it to, like, become active again?
That's a great question, and it's something scientists are still trying to figure out, but it seems like stress, sunlight exposure, even like hormonal changes can all play a role.
Interesting.
And another fascinating example of a provirus is HIV.
HIV. But doesn't that virus eventually cause AIDS?
Yes, it does, but what's interesting about HIV is that it forms a provirus but also produces small numbers of new viruses, even while it's integrated into the host cell's DNA.
So someone can be infected and spreading the virus without even knowing it.
Exactly, and this can go on for years without causing any noticeable symptoms.
Wow, that's a bit scary.
It is.
It really highlights how these proviruses can be like ticking time bombs.
Yeah, for sure, and it makes the development of effective treatments even more challenging.
Right.
Now, shifting gears a bit,
Okay.
I want to touch on something incredible. The fact that scientists have found ways to use viruses for good.
Wait, viruses for good.
Yes.
I thought they were the bad guys.
I know, right? It's a total plot twist.
How is that even possible?
Well, remember how they're so good at getting inside cells and delivering their genetic material?
Right.
Scientists have figured out how to harness that ability.
Okay, I'm officially intrigued. Tell me more.
So imagine this. You have a gene that carries the instructions for producing a beneficial protein, maybe a protein that helps fight disease.
Okay.
You can actually insert that gene into a virus, essentially creating a custom-made delivery system.
So the virus becomes like a tiny Trojan horse sneaking the good gene into the cells?
Exactly. The virus carrying its precious cargo infects the target cell and delivers the gene. The host cell then starts making copies of that gene and therefore the beneficial protein.
That's incredible. It's like turning a villain into a superhero.
I know, right?
Do you have any real-world examples of this?
Absolutely. One of the most well-known examples is the production of human insulin.
Oh, wow.
Scientists were able to insert the gene for human insulin into a virus, which then delivered the gene to bacteria.
Okay.
These bacteria then became little insulin factories, providing a reliable source of insulin for people with diabetes.
That's amazing. It's like we're learning to speak the virus's language and use it to our advantage.
That's a great way to put it. Now, before we wrap up our deep dive, I want to introduce you to another type of infectious agent that's even simpler than a virus, but just as fascinating, if not more so.
Simpler than a virus. I can't even imagine. What could it be?
Have you ever heard of prions?
Prions. That name rings a bell, but I can't quite place it.
Well, prions are nothing more than rogue proteins.
Wait, the proteins? Like the things that build our muscles and help our bodies function? How can proteins be infectious?
That's a question that puzzled scientists for a long time. It turns out that prions are proteins that are normally found in our bodies, but sometimes for reasons we don't fully understand, these proteins can misfold.
Misfold, what does that mean?
It means they change their shape, kind of like origami gone wrong. And this change in shape is what makes them infectious. Imagine one misfolded protein bumping into a normal protein and causing it to misfold as well. It creates a chain reaction.
So it's like a domino effect of misfolded proteins.
Exactly. And unfortunately, these misfolded prions can clump together and cause serious damage, especially in the brain.
That's a bit scary. What kind of damage are we talking about?
Well, think about diseases like Creutzfeldt-Jakob disease, CJD, in humans, scrapie in sheep, and mad cow disease, BSE, in cows. These are all devastating neurodegenerative diseases caused by prions.
So a simple change in the shape of a protein can lead to some pretty serious consequences. It's incredible and a little bit unsettling.
It really highlights the complexity and vulnerability of biological systems.
Yeah, it really makes you think like these tiny proteins, just by changing their shape, can cause so much damage.
It is pretty remarkable, isn't it?
Yeah, it's a good reminder that even the tiniest things can have a huge impact.
Absolutely. Speaking of impact, I think it's time we circle back to viruses and dive into one more fascinating aspect. Their genomes.
Genomes, isn't that just a fancy word for their genetic material?
You got it. It's their DNA, or RNA, and it acts like their instruction manual, the blueprint for building new viruses.
Okay.
And you might be surprised to learn that viral genomes are incredibly diverse.
Diverse? I thought DNA was DNA and RNA was RNA. What makes them so different?
Well, for starters, some viruses have DNA genomes while others have RNA genomes.
Right.
And within each of those categories, there's even more variety.
Really?
Yeah. Some genomes are single-stranded, like a single thread, while others are double-stranded, like a twisted ladder.
Huh.
And to make things even more interesting, they can be linear or circular.
Wow. That's a lot of variety. Why is this diversity in their genomes important? Does it affect how they behave?
Absolutely. The type of genome a virus has can influence how it interacts with the host cell, and even how quickly it evolves. For example, RNA viruses tend to mutate much more frequently than DNA viruses.
Yeah, mutate, you mean like change their genetic code?
Exactly.
But why is that?
Well, think of it this way. DNA replication has these built-in proofreading mechanisms like a really good editor RNA replication. Not so much.
Right.
It's more prone to errors which lead to those mutations.
So it's like RNA viruses are playing a fast and loose game of genetic telephone.
Haha. Yeah, that's a good way to put it.
And I'm guessing those mutations can sometimes be beneficial for the virus.
Exactly. Those mutations can lead to new variants of the virus, some of which might be better at spreading, evading the immune system, or even becoming resistant to antiviral drugs.
It's like a constant arms race between the virus and our bodies.
It really is.
They're always trying to outsmart us.
Uh,
Which makes me wonder, where did viruses even come from in the first place?
That's a great question, and it's one that scientists are still debating. Viruses are so different from other life forms that it's been hard to pinpoint their origin.
So are there any leading theories out there?
Well, since viruses can't replicate on their own, they must have evolved after the first cells appeared. One theory is that they originated as fragments of nucleic acid that escaped from cells and became parasitic, relying on other cells for replication.
So they were once part of a cell, but then decided to go solo.
That's the idea. And over millions of years, they evolved alongside their hosts, becoming increasingly specialized and adapted to their particular niches. It's a fascinating story of coevolution.
It really puts viruses in a whole new light. They're not just these simple disease-causing agents. They're complex entities with a long evolutionary history.
Right.
And they've played a significant role in shaping the world we live in.
Absolutely. And the more we learn about viruses, the more we realize how much we still don't know. They continue to surprise us with their diversity, their adaptability, and their impact on our lives.
Well, I have to say, this deep dive into the world of viruses has been incredible. From learning why they're not technically alive, to discovering how they hijack our cells, and even how scientists are using them for good. It's been a real eye-opener.
I'm glad you enjoyed it. Remember, the next time you hear about a virus, don't just think about the sniffles. Think about this intricate, microscopic world full of complexity, diversity, and endless possibilities.
Exactly. It's a world that's always evolving, always surprising us, and always reminding us that there's still so much more to explore. Thanks for taking this deep dive with us.